yeah, so maybe we can get started. It's time. Yeah, so as we know, the developmental biology is an inseparable part of light sheet technology. Actually, light sheet technology has grown on it, and so it's important. They are kind of inseparable, and I will not dwell upon our Professor Upendra Nongthomba's CV. It is well known and scattered across the campus and various online platforms. So he is well known. That sense, I know probably Professor Upendra is one of the finest developmental biologists at the moment, who is exploring zebrafish and Drosophila biology, specifically, which is relevant to slide sheet. So, I'll just go on to say that he will enlighten us with the biology of these organisms. And, and we all light sheet scientists are looking forward to address these pressing, biological questions, that are prevalent in developmental biology. Okay, so let us hear from him, and I think we're looking forward to. It's over to you, Upendra.
Okay, thanks, Partha. First of all, I mean, for giving me opportunity to share it. And I was just looking at the all the speakers in the last few days, and I mean, it's amazing that the technology development which people have spoken about. And I was wondering what is that I should be adding into. But anyway, so if you look at this title itself, we are talking about say cell imaging, live cell imaging technology. I have talked about here, put the title, but believe me, I'm not going to talk anything about the these technology aspect, because in the last few, those who have spoken in the last few days, I think they have covered extensively. Okay. So even though I'm going to make it very simple talk, so that you should look at it from the scientific development as a scientist. I mean, what question we unanswered questions are there that one should be excited about. See, two things can happen. One is that the two way of looking at the things. I think Partha belong to the first one, where you develop technology and look for the applications, right? So where it can address the questions, and that is the one way. So you develop the technology first, and then look for the how much advancement that technology can address, like say computer, or the say supercomputer and everything. So that is one way. The other way is that you look at the one specific problem, scientific question, right, and you want to address it, and you are looking for the technology advancements which can be utilized. And I belong to this second one, where we are trying to address developmental paradigms, right? And I have been trying to collaborate with all the amazing things. Like if you look at, we have used a different kind of microscopy. I mean, simple compound microscope, stereo microscope, to say electron microscope. And today we are using the light sheet microscope because of say Partha and Suez Kumar from IISc Trivandrum. And thanks to Partha, I think that we were one of the first to use a model organism to look at the whole organism using the light sheet microscopy. Uh, and if you look at, we are also using cryo-EM and things like that to look at the how exactly at the molecular level things work. And this has been extensively talked about, how microscopy has has been, I mean, the microscopes over the years, people have made microscope so that you can address from the looking at the from the micron level at the molecule level, single molecule level till the whole organism. And the challenge has been to look at the, I mean, the whole organism development, because whatever said and done, seeing cells is easy because to look at the how the cells, I mean, behavior, how they behave and how they divide and everything. uh, it has been easy, but the challenging has been the, I mean, how to look at the whole organism level, because at the organism level, there will be crosstalk between the cells and the I mean the organs, and when they form the organs, you know how they interact with the environment, everything. I mean, it's very, very dynamic. I mean, it happens at the cellular level also, but the cellular level, it is quite basic. Whereas when you look at the organismal level, it is quite complex and dynamic. So the how we can address that, and uh, uh, few areas where one can explore in coming years. So what is the most important question to developmental biology is that these single cells, right? How it developed to the full organism, say like me, right? And if you look at any adult person will have anything between say 120 to 170 trillion cells, right? All these trillion cells will have the same kind of genotype, right? Gene component will be the same, but then, right, we are not a mass of amoeba with a similar kind of this. I mean, we could have been trillion, mass of trillion cells, right? Just similar. But if you look at, we have specific number. Our hands are there, two hands, two eyes, one nose, hairs at a specific place, and head, then the thorax and the legs, etc. Right? Okay. This is what exactly developmental biologists are excited about, right? How these things grow, right? If you look at it, my hands are proportionate to my body size, right? It is not very long. I mean, um, so how do cell know that, yeah, now the tissue or the at the organ level, how do they know that there has to be some limit, right? Okay, what we call as the proximal distal or the dorsal ventral. If you look at in my front side, this is ventral side, okay, and my back side, yeah, these are all different. Okay, so how do these things at the what level when the cells are dividing, how at what level they know that there has to be some difference, right? That is the exciting thing. That is exciting because if you look at your hand, all the five fingers are different. It developed from a web like structure, right? And up to some extent genes expression matters, but ultimately it is about what are the mechanical things happening, right? Gene expression in collaboration with the the mechanical forces happening there, right? All those will decide which will become, say, leader cells and supported by the other cells. Then the surrounding cells, how they start, because even in your fingers, the cells which are in between, they need to die so that you have a finger. So when does that happen? slides?
Yes. Yes, it is visible.
Okay, thanks, Partha. Can you see it, right? Yeah? Okay. Hello? Yeah. Partha, you can see,
Yes,
hear
the
me?
screen is visible.
Okay, thank you very much. So the idea is that how the patterns are formed and how the cells they differentiate to different lineages. All these are exciting, right? Let's not dwell into it, but what is more important is that in the whole this process, it requires the movement. The movement is at the cell level. When the cells form the tissue, there is a movement at the tissue level, and there is a movement at the organ level. I don't have to give you example, but you need finally the movement at the organismal level so that we have the all this function properly, right? If you are unable to walk, you will not be able to do multiple things. That is a simple logic, right? So we are, if you look at simple example, is product of the movement. The sperms, they have to move towards the ovum. The fertilized ovum, they have to move on the fallopian tube, we call it, okay, on cilia, right? These are the cilia. I had movie, but I can't show you. So that these movements allow, we are product of this initial movement, right? What is the dynamic? Once you have the movement, is the dynamic is that the ovum is a single cell, fertilized embryo is a single cell thing when it is moving initially, right? So the when it moves, if there was no cilia to carry it forward, right? The movement of the cilia allow it to move in the one direction, right? Okay, so the movement of the cilia is directed, right? In the same way, many single cells, they like say amoeba or the protozoans, those which are single cell organism, or even the single cell, if you look at the neurons, they move by protrusion of the what we call as the leg-like structure, called as the pilopodia or lamellipodia and things, right? So the single cell move, right? Once, but you see, once it start moving, it could be because of this say food, which we call it a chemical cues, or the say electrical cues. Any kind of environmental cues can allow trigger the movement of the cells, right? And the cell also can move in terms of the say in the uh, uh, a group, right? And I will give you a few example how the cells move in the group, right? And this movement in the group is also important during the early development. And this movement of the multi group of cell is found in a stage of developmental biology called as the gastrulescence. That is the second, after the blastula, there is the gastrula stage. That is the second stage of major cell development. Okay, let's not remember anything. I mean, don't worry about the scientific jargon. But what I'm trying to say is that the cell can move in the single or the say multiple, but every cells when they move from, they go from say one place to another, say from point A to B. The point A to B, when some cell is not reaching from point A to B, then you can say that there is a problem, right? But if that can be studied by say snapshots, by taking at the point A or the point B, you can do it. But if there is a way to follow it, right? As if I'm going from say Bangalore to Mysore, the first thing what you need is that am I going alone or am I going with a group, right? Okay, because if you take a snap for me at Bangalore and if you take my snap in Mysore, it will not tell whether I have gone with my friends or somebody, my girlfriend, my wife, or anybody. You will know it, right? That can happen, right? Okay, that dynamic you can follow it only when you are following me with the camera, right? That is the thing. So when you want to go towards the mature, then you need a direction, right? For the every cell to grow and move around and think, you need directions. That I could have gone to towards Hyderabad. Instead of going to Hyderabad, I need to find a Mysore, and that is where I need direction. And this direction are helped by multiple signals, right? What we call a cell signaling cascade, cell signaling, what you call a traffic light, in other words, right? Okay. Or one way is that I could be just a passive diffusion which happen, but most of the time it is the active uh, transport which happen. And the active transport also, there are variables, just like in the, say, I could go to Mysore by cycle, or say by bike or the car, or depending on the whether I'm carrying a cargo, then a big cargo or a small cargo, I can go by train or the truck. All these matter. And same thing matters in the organism level, right? Okay, so we need to understand that those cargo, we need to understand the kinetic, we need to understand the mechanisms, we need to understand the traffic lights, which are the proteins, the signaling molecules, everything. And if I'm not reaching Mysore, then why I'm not reaching, what is happening, accident or something in the Ramnagaram, it could be accident with me, or accident something blockage by the other people, right? All these things dynamic you can study in the live cell stage, right? And once you understand that dynamic, what is wrong in the development, right? All most of the diseases, including cancer, most famous cancer and everything, is the developmental defect, right? Okay, development disease caused by developmental problem, right? Okay, so if you understand this dynamics, the process and the whole thing, then where to intervene become easy. The problem at the moment is that nobody has studied the actual stage when a disease happen, or just before the disease phenotype onset happen, right? We call as the etiology, right? That stage, if we are able to find, for example, say today, by simple one PCR, we can detect Alzheimer at least 30 years before actually the Alzheimer kicks in. But how many will go to take for the Alzheimer that test? I mean, right? That is the question. But that is for the different talk, right? So what I'm trying to say is that in the developmental biology context, we want to catch hold of a point where the 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 two cell, when they become divide, when they become two or four or eight, when they will define eye or ears or the nose or the hairs, all these specifications happen. And because if there is a defect tomorrow, we can intervene and say that this is the stage where you have to correct, so that whatever the defect is there, you can cure it. So that is in. And so
Upendra,
yeah.
just to say that your video is off. Sorry.
Yeah, that's fine, right? So that's okay. You want me to put on the video?
It's your wish. It's your
Right.
wish.
Okay. That's fine. So there are many people, those who have studied extensively, included in the diffusion. And the diffusion, you can't have it, say, for the long distance. That is why you need the active transport. And I know nothing about these equations, right? Okay. So people like Partha, or the people, those who have taken extensively the maths and the physics, you can calculate how much energy is required for the active transport, how much, because diffusion is not that energy expensive, whereas active transports are, and the cells need to be, conserved. And over the evolution, the whole body is very, very efficient. It is about the efficient usage of the energy. Okay. So I'm not going to talk about all these active transport, or the, say, diffusion equations and everything, how much they can. Just I gave you this one example, that depending on the size of the, you look at the animal cell, the size is numerous, and because of that, their movement will be very slow. So many of the active transport in organism is done by the motor protein, and the motor proteins could be either myosin or the say dynein kinesin family, right? And the myosin move on the what we call as the actin microfilaments, and the dynein and kinesin on the microtubules, and all your protein and all many of the cell up to this whole cell can move because of the dynein kinesin and the myosin. And because that you have different size of the cells and tissues and things, so the motor protein myosin or the kinesin family, they are huge, right? They have different size and different movement and things like that. Okay, let's not get into it. So ultimately what we want to know is that how we evolve. And if you really want to study it, because you can't, I told you, you can't kill human on thing, so you need to look for the model organism where you can really dissect it. And the life cell, whatever said and done, you need, way to look at. And the people, I mean, in the last 3 days people have talked about how you can label fluorescence and things like that. I feel that one is the technology advancement, where you are talking about the advancement in the microscopy and the imaging technique. Another way is that how you can develop, say, labeling in things like chemistry. I'm talking about the green fluorescence markers, or some other dye which can change according to the acidity, or according to the cell division, and or change in the environment. That is what it is more important also, besides the development of the microscopy. But people also have been working extensively, like say, ultrasound, for example. Say the whole development of the human can be measured without any labeling, right? So maybe Patha must have spoken to you about the, next challenge will be like, without labeling the cells, how we should be able to see it. And the classical example is the ultrasound, right? We are able to see it, but the problem with the ultrasound is that you can look at the whole gross development, but you will not be able to study the single cell, or at the tissue level, right? So if tomorrow somebody develop that kind of physics, right, that would be beautiful, right? And so remember that most of the tissues, organ, or the genes are conserved across organisms. So if you really want to understand even the human perspective, the disease or the development, you can study it by using model organism. And your choice of model organism is based on the question which you want to address. For example, say if we want to address the muscle is highly conserved tissue. So that you can even study the worms, C. elegans we call it, or the say Drosophila or the zebrafish. All these model organism muscle is highly conserved, genes, proteins are conserved, so you can study at that level. Okay, so you don't have to really study in the mouse or the even say a monkey. You can study it, you can find out what exactly how the muscle assemble to give rise to such a efficient tissue or organ, right? Okay, and if there is a disease condition, how you can develop drugs to intervene it. All those things can be studied. And the most importantly, if you want to look at the whole things, I mean, these are the organisms which are more transparent, which is genetically you can manipulate it. This is very important, because genetically any normal thing, how you want to know normal thing is that the either you have way to look at the how normal things develop, or you generate abnormality so that you can find out that the normal things happen because of the this if there is a defect in the normal things, you can have the abnormality, what we call in genetic terms the mutants, right? You generate mutations, you study mutation so that you can figure out how the normal things work out, right? So the model organism is a non-human one species which you choose to study a specific, say, paradigm in development, and which you can address it. Let's not get into too much, but so that you can choose today, you can choose any kind of model organism based on your interest and thing. And but particularly being in India, you have to choose a model organism which is not very expensive. For example, say if you want to generate Drosophila mutation, it just doesn't take much money, like say 36,000. Whereas if you compare the same kind of transgenic, if you want to generate it in mouse, it will cost you anything between say 4 to 5 lakhs, right? Yeah, and it takes longer time, right? So, and you can get all these mutation from the Drosophila stock centers also just for $26, right? Whereas if you need to get it from the mouse or for the even the zebrafish, you need to spend at least like say $1,000, right? Okay, so just being where you are working, it also matters a lot, right? And most importantly, ethical clearance. At least in India, you don't need the ethical clearance to work with the Drosophila or zebrafish, whereas you need the ethical clearance for to work with the mouse and higher organism, right? So we have been using extensively Drosophila for the early development. I'll just show you a few examples why we get excited with the whole thing. And the most importantly is that the Drosophila, I told you that you can generate mutation, you can generate abnormality, and so it is very easy to follow the whole thing, right? You know what are the genes which are required for the development of the specific organs or the say tissues, right? Okay, but what people have generated mutation will give you that for this, I mean, if you have a mutation in a specific gene, you can't have eye or leg or say wings. But that doesn't give you that how exactly those gene products are required for the dynamic development of these organs, right? That is what the microscopy live imaging can do. And these genes have been like identified for long time, and from long I mean, these three people particularly identify almost like 60,000 mutations, and they identified that these genes are conserved till human, and for that they were given the Nobel Prize. And this is one of the uh, they got Nobel Prize in 1995, and that was the one of the reason that I got motivated to work with the flies, because I completed my master's in 1994, and I started my career in Drosophila after these people got the Nobel Prize. Okay, so seeing is believing in that is what I'm trying to say in Drosophila early development. For example, say if you look at the position of the nucleus is very, very important. Now these nucleus start growing from initially it will be here, then they need to go to here, and the movement of this nucleus will decide whether this region will be say dorsal, and this region will be ventral, or this region will be so anterior, this region will be posterior. In other words, the axis or is decided by the nuclear movement, right? For example, right? Okay, let's not get into the molecule now. When the nucleus move, the question come is that, right, whether the nucleus move there by diffusion without anybody's help, right? Second thing is that if it is taking help, then it is carried over by I told you, the motor proteins actively transporting it. If it is actively transporting also, then you can say that whether it is being pulled or being pushed, right? Both are possible, right? And this is where people met live cell imaging, and this is where they were early development embryo, and they were able to show that actually it is the pushing rather than the pull. So for you think about it, the Drosophila development people have studied for years, and this paper came out only in 2016, right? For so many years, Drosophila has contributed so much. And the oocyte development, people have been working left, right. People have identified, the mutation where this nucleus, right, doesn't go in the right place and there is a defect. But people didn't know until the life cell imaging that how exactly this nucleus go from, say, posterior region to the anterior region. Okay, and this was possible because of the live shape imaging, and I can't show you the movie. Okay, it was a huge file and it didn't work, right? Okay, so the people were able to exactly show it by pushing it, right? I mean, by looking at the live imaging and showing that it is actually push, right, rather than pull. Similarly, we have a very well-known thing called as the epidermis to epidermal cell, like the hexagonal cell, to not hexagonal, the like tubular kind of cell, epidermal cell, how they move to elongated structure like called the mesenchyma. And this mesenchymal cell are the one actually which are responsible for metastasis in the cancer condition. And this is a big question because you need to have it for the epidermal cell like to mesenchymal like, and I mean, in other words, the cell need to change in the shape, right? Once the cell shape change, they move to different place, and after reaching that second side, they again change to epidermal like so that they can grow. And this has been studied extensively using Drosophila as a model. You can see that these cells, they shape from here and they migrate and they go here, and again once after the migration, uh, it looks like the cell, there is no change in the cell shape, right? Okay, because the cell become like this structure after reaching here. But only when people look at the by people look at this migration by live cell imaging, you will appreciate, you will appreciate it only when you see it. You can see that how cells are trying to come out of that layer, right? Say 8 cells, they move away. You see the dynamic here undergoing, how the cells are moving. Now, just by looking at it, right, you can have so many questions, so many, like that. How the cells know that the only 8 of us we have to migrate, right? First of all, it could be signal. Then once they are migrating, right, they are moving. Which cell is the leader cell? Right, some cell has to be leader to pull it, right? Okay, so you can appreciate this again in this movie. Okay, you see it's you, but the changes, it changes, the leader changes and the work around, they crosstalk with the these cells, neighboring cells, right? This is from the confocal live cell imaging, but I would like to see this in the 3D. That will be beautiful, right? Isn't it? 3D, how the cells are organizing, how they are changing in the shape. And that is what Partha can solve with me in coming years, right? Because the crosstalk with the cells are these niche cells we call it, are also important, because otherwise they would have not been able to move. And we have identified mutations where these actins, we call it cytoskeleton, will not allow these cells to move to its destinations, their destination. Okay, so this is what I'm excited about, development, because otherwise development will not happen. If it doesn't reach here, development will not happen, right? And all this migration happened because of the myosin, and which myosin is involved, myosin 6 and myosin 5, all these are involved, and let's not get into that, right? Okay, and movement of the cells also is required. For example, this movement of the cell is also required for the development of the whole organism, and this can be stopped any place. And once the cells move, they have to be enclosed, and this is what we call as the dorsal closure. and these cells come jipping, and it is used as a wound healing model. I mean, right? Okay, it just work like the what when you have the injury, right? Okay, now the question come is that what are the molecules which comes near to this is what exactly we are trying to study, and this again can be studied by live cell imaging, and but most of the live cell imaging, as I told you, it has been 2D, and looking at the whole thing, I would like to really see what happened there, where from the macrophages of the, the, in other words, the dead cells need to be removed from there, and what are their role and everything. I mean, we need to study that in future. The same thing applies to the neuronal development, and I'm excited about the neuronal development because neuronal development happens with in conjunction with the muscles. I mean, you can see that neuronal development is dependent on muscle. I will show you, but so this need to grow, but no, people have shown it in the cell culture, but people haven't shown in the in vivo, inside the organism, that the, how this neuronal growth is dependent on, because they are restricted by the neighboring cells, the cues coming from within the cell and the outside the cells, right? So how they crosstalk this, these are not known. And within the cell itself, the migration of the, say, many ATP producing mitochondria and all these matters, and the movements are hampered. That is where you have Alzheimer, Parkinson's, and all these diseases. Okay, so for the movement, again, you need a motor, and let's not worry about it. So I'm interested in the muscles because the muscles, you can follow that, you can see that this myoblasts, which are the single cells, they move, fuse, and they develop into these wonderful muscles, right? Okay, and most of the diseases in the world, 10% of the movement disorders are because of the abnormal in the muscles, right? Okay, and we have been trying to study this in vivo, right? The challenge has been to look at the in vivo, how assembly happens, and this is where we have taken help from Partha, and thanks to Partha, that at least we have been able to look at the Partha and the Suez from IISc Trivandrum. We have used the light microscopy to look at the whole thing, that how the muscle develop, and how the muscle crosstalk with the, say, other energy, because they require the energy like the fat cells, because if you remove the fat cells, then the muscles will not develop at all. So the muscle development is based on the fat cells and the neurons, and how they crosstalk and things. This is what exactly excites us, and this we have studied extensively with the light microscopy at the moment. But we need to address this with more what you call magnifications, and how they become so symmetric, right? All these things need to be addressed in future. So the muscles, I told you, neurons grow based on these muscles, and you can see this dynamic. All these are snapshots, right, from a single, without any neuronal growth. Now you can see all these branches, and this branching, how they grow. These are a snapshot, but what happens in the dynamic, that is what we want to look at in future, right? And similarly, Drosophila will have only one copy of the genes, whereas we have multiple copy. So we have used extensively zebrafish also as the vertebrate models, because vertebrates are different from the invertebrate like Drosophila. And you can see here, this is intertoto microscopy. Every cells were tracked, right? Okay, you can see it here. Can you see the movie, Partha? Because it was a huge file. Hello?
Yes, sir, it is visible.
Yeah, so this is taken from EMBL website, and you can see that, I mean, this is amazing for personally for me. This is what I want to look at with help of with Partha and other people. I mean, you look at when the cells are developing, the cells, how they divide,
asymmetrically and move all around. But finally, they assemble in a specific place to form the embryo, right? Isn't it beautiful, right? So now the question is that out of these cells, what decides that the means in the embryo that they have to be on the anterior sides? If you look at it, it is kind of random division, but it is not, right? So you look at it, some will be anterior, some will be posterior, and from all these things, now the question comes is that which will form the eyes, which will form the muscles, because the one which are here in the, they will form the muscles and the neurons, and the brain will be here, right? Okay, on the I'm sorry, pointer is not there. So the brain will be here and the muscles will be here from these cells, right? So they were able to show it in the early embryo, but exactly what happened in the later stages, that is the challenging, because you know we need to follow for at least like 72 hours with the specific organ in mind, and we have to do it, right? Yeah. So similarly, at the cellular level, I'm unable to show you the movie. Cellular level, when you get infected or you get injury, the macrophages, right? They move to the infected site. This is a beautiful movie. You can see it on YouTube, right? Okay, and this is what we are developing at the moment in ourselves in the lab, where we want to look at this macrophages, that the dynamic of this macrophages when you have injury or you have a heart attack, where the cell deaths are more, how they are removed. So, and what drugs, once you know the molecular thing, that how they come, and can you make them come faster so that the injury is closed better, or they say how you can enhance the removal of the dead cells, right? All these things matter, and this is so exciting because you need to follow this in the live animal. There is no point in looking at the dead animal. Okay, in the live animal you need to look at how dead cells are identified, how dead cells are removed, and what drugs you can use it so that you can enhance the whole process or stop the whole process. Okay, that is what we are trying to develop, and this whole process is what we call as the efferocytosis, and my postdoc Sumit is making all these lines, and hopefully with the help of Suez and Partha, we will be able to address that, right? Similarly, we can look at the what happens, when you suffer from say uh, what happens when you suffer from some disease of the cardiac arrest or things. And this is what we have now, all these GAP level geographies, and we will be able to follow this in the live way, and we will be able to screen the drugs, which will uh, intervene all this dynamic, right? Okay, so I'm just excited about the whole thing, is because there are so many unanswered questions. The kinetics of the, say, most of these cells, right, when they move around, what happened and things. And luckily, I have Partha and the Suez. They are helping me, because you can see it. This is the light microscopy which Suez took it, and it is published. You look at the zebrafish, the whole body, right? Each septa of the muscles, they are so symmetrically organized. They start with asymmetrically. And recently there has been a Nature paper from the German group where they showed that this is because symmetry is developed afterwards. Now the question is that, we were in the similar thinking line, but you see, it's a highly competitive field, right? So before we could do anything, they have published, and they have just recently, last week, the paper came out in Nature. So the question is that, what are the things which are involved in making these symmetrical things, right? And they have shown that it is because of the mechanical property rather than the genetic property, right? Okay, beautiful, right? Okay, so this is where we need help from people like Partha, or the people, those who are developing new microscopy, to look at the, every septa, for example, developing, so that the abnormality, when people have the abnormality, we can understand when to intervene. That's the whole idea. And similarly, we have made a human disease condition in zebrafish. For here it is the eye development. So the eye development is dependent on the all these cells going proliferating and dividing and exiting at this right time. And this is what we would like to look in future, that right. Okay. And so we have many drugs which you treat it, the muscles or the tissues, they become abnormal, neurons become abnormal. All these, when the induction happen, this is what we want to look at by live imaging. When do the cell die, right? That is what we would like to look at. And we are now collaborating extensively with Akash Guliani. Akash is the person who makes this visible dye, right? Okay. And that dye is not toxic to the organism. And now, and those dye will change in intensity, based on the condition of the membrane or cell membrane or the mitochondria, and so we should be able to follow the dynamics, that what happens and what stays, including diabetes. Maybe say, for example, in the diabetic condition, when we should be able to say that you are prone to diabetes, this is where the cell is, and if you exit beyond that cell stage, then you are incurable. You will become incurable. But if you are in this stage, then you are curable. And that is what we want to find out by using zebrafish as a model. And I'm hoping that people like Partha will help me out, right? So what, in the nutshell, I have just given you simple few example, but what I'm trying to say is that I'm excited about the probability that brings by, say, for example, I told you, by the technology development at the one hand, the imaging technology, on the other hand, like the chemistry, the fluorescent, the probe, right, which can be done, or the other one which we are also collaborating with, is that the development of sonography condition for all these small animals, so that we can look at without actually using any probe, that we can see the sensitive. It will depend on the sensitive nature of the, say, organ and the things, like, say, heart we can monitor easily. But are there any instrument, can we develop any instrument, which can actually change uh, the bit of the, say, muscles or the thing, so that actually we can really work into it, right? And you can see that this is possible, but this comes from the cryo EM reassemble to make it as a movie like this. But this is exactly what I'm interested, is that what happened, right, at the in vivo condition. In vivo condition, what is happening you see, look at the steps involved in the muscle contraction. Yeah, okay, every steps is important. Yeah, so this is what I'm excited about, that how to follow these steps, right, so that we can intervene when somebody suffer from the disease condition, right? Okay, I stop here. And so the whole point, what I'm trying to also say, is that it will depend on the collaborations between different fields. If I want to address something now, I cannot do it without collaboration and help from people like, say, Partha, or say Akash Gulyani. Akash is a chemistry person. So I need help. And they also need to make sure that after the development of the imaging technique or the technology, what bigger question that can be addressed, so that we contribute to the growth of the science, right? Okay, so I stop here, and I would be willing to take any question if there is any. Thank you.
Yeah, thank you, Upendra. It was a wonderful talk, and I think it's amazing how an entire organism emerged from a single cell. So, what your talk has done, it has filled a vacant gap in our in our workshop, because biological perspective was something that was, I thought, was missing. It looks like developmental biology is very, very rich, and there is a lot to explore. Okay, now we're open for questions. So, if anyone wants to question, please come up with your.
I mean, I'm not expecting, because you people are technology people, but
Right,
if
right.
there is any clarification you want to have it, I mean, I
Yeah,
most welcome.
yeah, yeah.
Offline audience, any questions? Anybody? Sir, one student has one question.
Yeah. Yeah, please. Can you read it? Hello?
Yes.
Hello, sir.
Yeah. I
Okay.
can't.
I want to know that what is the time scale of the migration of cells or any other developments?
Oh, okay. So it depends on the animal that you are using. For example, say if you want to look at the dorsal claws, which I told you, right, in the Drosophila embryo development, it will be just for 6 hours, right, 3 to 6 hours. That's it, right. Whereas if you look at the zebrafish development, the whole embryo development completes in 72 hours, right? Okay, and the migration of the cells, again, I have given you the example that it depends on the the cell to cell, I mean, right? Okay, and whether it is moving as an individual or as a whole, many cells, right? Many cells, if you are looking at, they are always slower than the single cells, and it can vary from, say, few minutes to 2 hours. I mean, depending on the early development or later stages. In the later stages, it can go up to, like, say, 32 to 40 hours, right? So, for example, say neuronal cells, the myoblasts, which I talked to you, myoblast cells, they migrate within, I mean, initial 16 to 20 hours, right? So it will depend on the cell size and the cell dynamics, okay? But it can vary from anything from, say, few minutes to few hours.
Anybody has
Okay,
any more?
thank you, sir.
Yes, sir. One more question.
Yeah.
Professor, one question.
I can't hear it. Can somebody?
Oh, unmute. Oh, yes, thanks. I have one question. Just because I'm not from your field, but I just wanted to know, like you mentioned, like you can use PCR to detect diseases early. Can you just briefly explain how does that work, or what is the science behind that? Like, how do you use PCR to disease
Oh,
like
it is.
Alzheimer's
So
earlier?
in this condition, in the Alzheimer, for example, the minute, it is genetic, then it is easy. But the genetic basis is only, say, mutation can be detected anytime. Okay, from the baby itself, you can detect whether you are prone to a specific disease. For example, say my father had the diabetes, then I can test my gene by using, say, there are close to around 25 genes which are very highly correlated with the diabetes. I can sequence all those genes easily by using, PCR is just to make the DNA multiple copy. That's all. I mean, right? Okay,
Right,
and we can
right.
sequence it and we can identify it. Similarly, in the Alzheimer case, however, there is a expression of a gene which goes up in the our neurons, right? Okay, if you are prone to Alzheimer. Okay, so not mutation. It is not necessary to have the mutation, but if you are prone to, say, you are going to develop Alzheimer, then these genes go up much before almost like say 30 years before the actually the phenotype is seen. So if you take a few liquid from the we call as the CSF, cerebro spinal fluid, we call it. This is the fluid which is between the brain and the your skull. So you can take out few fluid from there, and you can just look at the expression of the gene by using the PCR, in the sense that the real-time PCR. Look at the gene expression, and you can say, depending on the how much is expressed, you can tell that whether you are prone to Alzheimer, whether you will develop Alzheimer in 30 years time. The prediction is at the 30 year is only around 67%, but 15 if you go up to closer and closer, for example, say before 15 year it is 87%, right? It means that the accuracy will be 87%. If it is, say, Alzheimer will develop in 15 years time, then so the 30 years is around 67 to 68. In other words, you may develop and you may not develop. But the good thing about knowing such thing is that now you can take the precaution, right? So say, if I'm prone to diabetes, if I knew that I am prone to diabetes by sequencing and everything, I would have never joined IISc, because scientists, diabetes happen because of the stress, and or most of the IISc professor, they suffer from stress, unknown stress, right? Student stress, or the funding stress and things, and you can suffer from diabetes. So I can tell you that the 80% of our professor in IIScs are all having diabetes problem. So in other words, what I'm trying to say is that you can take the precaution if you are predisposed with this gene expression and the mutations and things. All right, you can take what we call as the informed decision. Is that answer your question? Does that answer your question? I hope.
Yes, thank you.
Yeah, so I
Sir?
have one. Yeah, any more questions?
In chat box, one online audience has put question
Right.
on chat box.
Yeah, can you just read it so that I can answer it?
Yeah, I will read it. You talked about cell-cell communication. So when we culture a particular cell line artificially, is the growth of the cell sensitive to particular wavelength of light?
Yeah, this has been shown extensively. If you put different, even when the cells are, you are taking for imaging. Now today there are data which suggests that even the fluorophore will harm the cells, right? Different light, if you are putting it, whether you are putting even the UV, or the, say, you are putting visible light, or things, that the cells can sense it, right? And there are many sensitive cells. For example, say, people have shown even in the organism level that, for example, planaria and things, they different light will impact the way they sense they go towards the light. Okay, and so light is a cue which people have used extensively for the cell migration. And there is a beautiful paper which was published in PNS by Akash Guliani. If you are interested, you can have a look. It is on the, why we develop eye in the first place, was studied by using a simple planarian eye development, and it is beautiful, because even when they don't have the eye, they sense to different incidents of the lights. And so cell movement is based can be based on the light, or the, say, chemical, or some other things. Okay, even the electrical and things like that.
Right. More questions? Yeah, Upendra, I have a question. It's a curiosity. What drives the movement of nucleus from posterior to interior of a Drosophila? Is it random, or is it like there is some direction?
No, there is a direction, and the direction is because of the growth of the microtubules, right? Okay, because the microtubule growth has got the direction, right? You know that it grows from the positive to negative, negative to positive side. So it depends on the growth of the microtubules. But what is still a enigma is that in the why not randomly, in the sites? If you look at the growth, even when the cell is pushing, if you look at it, is going towards one side, not in the middle or in the other side, right? So what really makes it to go, because if you are coming from the center now, it can go any direction, right? And because microtubules are just going from the posterior to anterior. But what makes the nucleus to move to one side is, I mean, people have modeled it mathematically and everything, but believe me, that dynamic, that is what I'm looking into, is that if tomorrow, if we can have means way to level it, and we can look at it, yeah, 2 photon people have tried, but it didn't succeed. I mean, there is a way. That is where I'm looking at, is that tomorrow, if we can have a even though cuticle is little bit, darker, if we can make it light microscopy, and the image when the nucleus is moving, only thing is that you remember, the challenge is the resolution. That is why I was trying to develop the light microscopy with the 63x, right, with Suez, right? Yeah. You remember I was talking to you about it. So we are trying that, but it is a challenging thing. So the thing is that how exactly microtubule growth push the nucleus to the one side. This is still a question. I mean, right? But it has to be some signaling and crosstalk with the microtubules and the nucleus thing that needs to be looked into it.
Right. Okay, I guess we have another question from Prakash. Prakash, can you read the question?
Yes, sir. In the cell culture, we usually get contaminated because of the bacteria, and the cells die. Is it possible that we can like create some bacteria which can kill cancerous cells? Because bacteria usually contaminate cells and they die.
Oh, uh, not exactly bacteria, but at least for I can tell you today is that your gut, the colon cancer people, those who have colon cancer people have shown that they have it because your gut bacteria, though they don't produce a good enough what you call good metabolites. Okay, we call it as the bacteria also should produce nice compounds, amino acids, other required things, right, which you need it, right? So your gut microbiota matters a lot. You can, in the cell culture, the bacteria or fungi, they can kill your cell. Okay, contaminations, and there are many now cell uh, antibiotic resistant fungi and bacteria which can kill it. But when you put those, you need to realistically look at how you will put those bacteria in the every cancer causing cells, right, in body, right? Okay, that is the challenging thing. It is very nice to ask that question, but rather we should look at is that how we can make what is that in the bacteria which can kill the cells, right? Okay, some metabolites which the bacteria secretes, right, which you can now modify to make it as a cancer specific. See, the whole point in the cancer treatment is that you are unable to kill the cancer cell, targeting only the cancer cells. So the side effect which you suffer during the cancer treatment is because you have the off target of the drugs and the treatment strategies. Okay, so now we have antibodies, we have drugs which can be targeted into the cancer. But the problem with that is also is that the cancer cell, just like on your cell, when you start with a single cell, when you make it clonal cell, we call it, starting with a single cell. The moment single cell become two, they are different. Okay, just like in the human condition. So the when cell grow, they are different. Okay, they look like they may look like same. They are HeLa cell, but people have shown that when you grow the cells, they are heterogeneous. In other words, the property will be different. So when you put the drugs in the, for example, treatment of the cancer also, some cell will die very fast, but every drug has got the time to go flush out of the body, right? Otherwise your body will be harmed, right? So what happens is that some of the cancer, they are resistant to drug. That is the problem with the cancer cells. They develop resistance to drug. Now those, even one cancer cell, if you can't kill it, what we call as the cancer stem cell, then they can again grow, what we call as the relapse, right? Then you will have again the cancer again grown, right? So these are the challenging thing. So what you have observed in bacteria killing cancer cell is fine, but practically to do it, it is a challenging thing, because to in the body to find to infect every cancer cells with the bacteria, that will be challenging without the harming the other body cells. Okay.
Yes, thanks.
Okay. Any more questions?
Yeah,
Yeah.
Yes, so cell can form certain patterns with the external stimuli like laser of some wavelength. Is there any difference between patterning of cells with a normal cell and a cancer cell? Whether that there can be any difference while
Oh yes.
patterning?
Yeah. So the actually today we know that the cancer cells are actually, if you look at, I call it as a like developmental paradigm, because if you look at the most of the cancer cells, when you are developing during the early developmental stage, you need a set of gene to be expressed so that you develop as an embryo, right? Amazingly, it's all about the timing. Those genes which are required for the early development, now you find it expressed in the cancerous cells. Do you understand? So the most of the cancer cells are cancerous, mostly, which we don't know the exactly how they do it, is that most of the embryonic genes which are required for the embryo development, they can again switch on, right? In the adult stage, for example, all these embryonic genes are off. But in the cancer cells, what happens is that these embryonic cells are again switched on so that they can develop new blood arteries, so that they can absorb more nutrients from the neighboring cells, right? And they can grow as a whole body. If you look at the cancer, actually, it is a kind of a embryonic growth. Most of the cancer. Okay, solid tumors, if you look at, is the embryonic growth. They will have multiple patterns, like for the blood development, some other kind of things. They will have it, right? So and the other thing which I would like to take this only 1 minute, Partha, is that okay? Just to cancer, because
Yeah.
many
Go ahead. Go
many
ahead. Go
people
ahead.
are asking the cancer question. The
Right.
philosophy of the cancer you need to know is that whole body development happens because there is a synergy between, even when the patterning is happening, after the patterning, just the genes get switched on, off, and things like at the right time, at the right moment. So the cell sacrifice, I told you, so that you can have leader cells growing and the fingers like that. So your pattern, your nose are proportionate to your body, your eyes, and everything, is because the cells switch on, off, and the way they grow is highly controlled, right? But what happened in the cancer cell is that uh, the cell become greedy. The moment cancer goes, the cells grow and they want more and more, just like some of our leaders, right, or the corrupted people. They want everything for themselves, not for the society. See, finally, they are so greedy that they will completely kill the whole body. And when the body die, what happened? The cancer cell also die. That is why I said that everything in excess, it is bad, and particularly when you want to have all the grant money, this is for the scientists, all the grant money for yourself rather than for the other people, colleagues, it also is bad. And the same thing applies to people or the leader and political people, that they should not be cancerous people. All right. Yeah. All the patterning genes, different kind of genes are switched on in the cancerous condition. Thank you.
Thank you, Nongthomba. Any more questions?
So I have one question.
Yeah.
I'm not a biologist, but I'm curious to know like how people found some organs inside the cell. Say for nucleus is there inside the cell, DNA, RNA, all those things are there in the inside the cell. So how people initially found it is there?
Actually, you should look at some of this historical perspective, how people identify by simple, basic, say, DNA or RNA. They bind to a specific dye, for example. Okay, so the DNA and RNA was labeled with, for example, say simple chemical dye called as the fusin, which binds to the DNA and RNA. That is where they were able to see the, the DNA and RNA is highly concentrated in the nucleus, because when you stain with the DNA and RNA, you can see the nucleus, right? Okay, and this is how people identify all these organelles by microscopy per one, and at the same time, people, those who are doing the chemistry, I mean, those who are developing different staining protocols, right? Okay. And if you look at it, there are classical experiments by, I mean, how DNA even replicate, and, whether it is a conservative or semi-conservative or all these things, it's amazing. Just like, as I told you, you need to have the questions, then you can look for the instruments and development things. I mean, you can do it. At the same time, if somebody say Robert Hooke, when he developed the microscope, he started just looking at whatever leaves or anything, right? And he started drawing them, right? And that is how he was able to look at the cell within the cells, what are the things visible, right? Okay. And that is how many of the bigger organelles, like say mitochondria, they discover that in the there are cells where the mitochondria are really, really big, right? For example, the muscle cell, if you look at mitochondria, is huge. You need phase contrast and things. Up to some extent you should be able to see it, right? Okay. So there is a amazing history to the discovery of all these organelles and things, centrifugal methods, the chemistry, staining methods, then microscopy. Microscopy, I'm talking about not only about the simple microscopy. For example, say mitochondria cristae was visible only when people started looking mitochondria under the whole cell under the electron microscope. If we would have not invented electron microscope, then we would have not appreciated what are the things available inside the mitochondria. And if we would have not invented cryo-EM, then we would know many of the structure at the micron level, right? So the angstrom level, some of the proteins, if you look at their structure, have been solved by, of course, people use many different kind of X-ray diffraction and all these things, but today the whole structure, complex structure, can be seen by using cryo-EM, right? But for that you need a technology. But once you acquired, I think Partha must have explained it, but it is not only about acquiring the images, but how to resolve those images, right? You need many image processing things you need to develop, right? so that you can see them properly, because still there will be many ways you can see the things, right? Okay, so you need to be expert now today in if you are technology person. I know that you people are not biologists, but you need to be good in imaging development of the technology and things like that. You see, I did a postdoc with Professor Justin Molloy. Justin Molloy is a person who developed single molecule optical tweezer microscopy. So the James Pudick and Justin Molloy was the first to develop the optical tweezer. I think Partha must be knowing it, right? Okay, to look at track the single molecule within the cell. Okay, and I work with Justin. I mean, so I started my career with the single molecules, how the actin and the myosin they interact. Okay, but only thing is that somewhere you need to find your needs, and I was very bad in understanding the kinetics. I mean, because all my physics and maths were mug up and wrote in the exam. That is the bad thing about the Indian education system, right? So and I didn't take physics and maths afterwards, so it was very difficult for me to appreciate the thing. That is why I moved to genetics, which I'm comfortable with, right? So you need to find it, but I have seen it. A student of Justin's, Gregory Mahasev, right, from Russia, will dismantle microscope, the whole optical tweezer microscope, within 3-4 hours, right? Because the pinhole was not correct, right? So he will completely dismantle microscope to pieces. Then he will assemble again, taking close to like 12 hours, right? Okay. If you ask me to do that, I will give up science, right? So it is up to you. What are you excited about? Where you get a motivation to do science, and that is what you should follow. And that much I can answer.
Yeah, thank you, Upendra. I think if there are no more questions,
Thank you.
yeah, then we can end the talk. And it was a great talk. A lot of things that we have learned, and it has given us the biological perspective that we're looking in the workshop. And thanks a lot for all the audience who has actually, pitched in. And thank you, Upendra. Thanks a lot.
Okay, thank you very much.
Yeah, yeah. Thank you.